This is an interesting topic. I'm not at all an expert on soil microbiome, even though I've been harping a little bit about the importance of soil carbon as a part of increasing soil health, its retention capacity for soil moisture, nutrients that go with it, and so on, and methods like agroforestry which enhance soil carbon. And here in IIT Bombay campus, Arnab Datta is trying to lead a proposal on, you know, sequestering carbon into the soil and so on. So, and I have a course on sustainability. So overall, I think paying attention to soil carbon as one of the carbon pools that's important for climate mitigation at some level, but it's much more important for crop yields, farmers' income, and overall health, biodiversity of the system, and so on. So this paper is a review paper from Nature Biotechnology. So Nature now has so many franchises. You have a Nature journal for every topic. So soil microbiome engineering for sustainability in a changing environment. I will just read and try to say some things about the things I can't say something about. It's very technical as well, so you have to follow up on the details if you're interested, because I'm as usual going to be very brief. These are just meant to introduce you to the topic where exciting research is happening that I find useful for climate science, climate change, sustainability, adaptation, and so on. Recent advances in microbial ecology and synthetic biology have the potential to mitigate damage caused by anthropogenic activities that are deleteriously impacting Earth's soil ecosystems. Here we discuss challenges and opportunities for harnessing natural and synthetic soil microbial communities, focusing on plant growth promotion under different scenarios. We explore current needs for microbial solutions in soil ecosystems, how these solutions are being developed and applied, and the potential for new biotechnology breakthroughs to tailor and target microbial products for specific applications. We highlight several scientific and technological advances in soil microbiome engineering, and including characterization of microbes that impact soil ecosystems, directing how microbes assemble to interact in soil environments, and the developing suite of gene engineering approaches, genetic engineering. This review underscores the need for an interdisciplinary approach to understand the composition, dynamics, and deployment of beneficial soil microbiomes to drive efforts to mitigate or reverse environmental damage by restoring and protecting healthy soil ecosystems. That's the overall idea of the review, with many details and a very nice long list of references. So here is a nice schematic of soil microorganisms that carry out key ecosystem services and have the potential to help mitigate a variety of deleterious anthropogenic impacts on the ecosystems. White box text indicates different deleterious anthropogenic impacts, and yellow box text indicates different biological mitigation strategies. The rhizosphere is highlighted in the circle in the center with associated mycorrhizal fungi in the green threads, nitrogen fixing nodules forming bacteria in the brown nodules on the roots, and a variety of root-associated microorganisms that are indicated by the pink and yellow symbols, top left. Heavy metal gray hexagons, so you can see these here, that can be bound by heavy metal binding bacteria, as seen as red rods here, and you can see them bound already as an example. And pesticides in light green dots, so you want to remove those things that can, pesticides, light green dots that can be degraded by rhizosphere microorganisms. So these are like a nature-based solutions, except you are doing some soil microbiome engineering. Bottom left, potential pests that include viruses in dark green, like here, and bacterial blue rod symbols here with the tail, and nematodes with green ribbon. Carbon, nitrogen, and phosphorus are shown, of course. So you have things like earthworms, which are the which are part of the healthy ecosystem that are getting damaged by human perturbations, including warming, pollution, pesticides, and so on and so forth. So you get the idea. You have heavy metal contamination and heavy metal binding here with some viruses and pathogen resistance. So you have pests and pathogens working together. Biopesticides induced systemic resistance to pests and pathogens. And you have pesticide contamination, so you do pesticide degradation with some bacteria, again soil biotechnology kind of thing. Over fertilization is a deleterious effect, nutrient runoff, salinity, drought, low organic carbon, nutrient limitation, etc. Somebody is standing there and shouting nutrient turnover. So these are the mitigative actions that are imagined. Osmotolerance, drought tolerance, carbon sequestration, nitrogen fixation, phosphorus acquisition, and micronutrient acquisition. So nutrient limitations in phosphorus and nitrogen macronutrients. So you don't want to necessarily just depend on fertilizers. This is talking about nature-based kind of solutions again. So here you have carbon fixation as well. So sorry, I tried to hush the guy, but that doesn't work. The three that's going to be in the podcast, so sorry about that. The three pillars of soil microbiome engineering are isolate discovery, assemble the communities, and genetic manipulation. The interplay among these pillars will lead to advances in providing soil ecosystem services, including mapping microbial function to organisms and genes, improved ecosystem functions through the establishment of naturally evolved and synthetic microbial communities enhanced by engineered isolates, and thirdly, promoting biosafety and biosecurity goals by understanding colonization and containment dynamics at ecosystem scale. So you have isolate discovery, soil microbiome engineering is then feeding back with assembled communities and ecosystem services on that side, and you have genetic manipulation for isolate discovery and assembled communities as well. Being very kind of brief, but just want to give you the idea. Design of tractable communities of soil microbiome. So you're talking about real engineering here. So this is a naturally evolved root NatCom, complete soil microbiome with microbial interaction network here. What are we doing? The design of tractable communities usually proceeds along one of two routes. In the naturally evolved route here, NatCom, naturally interacting members of the soil microbiome are enriched from soil. In a SynCom, individual isolates are combined to form a community. A SynCom leads to much greater control of the community's constituents as well as more inherent knowledge of the potential processes, but the individual SynCom members may not naturally interact and may be less stable in the environment than a NatCom. So these have evolved over time, and these you are doing through technology, biotechnology, and you don't have time to see over time what will happen to them, right? Reduced complexity community and SynCom is over here, and again you're talking about pathogen resistance, bioremediation, and drought resistance, and you want to organize microbiomes and worry about their interactions. And of course you're always worried about whether this is all a Frankenstein experiment or if it's safe in terms of biosafety and biosecurity, right? You don't want things to go out of hand. Positive, so here we are looking at schematic of a co-abundance network for microbial taxa. Microbial taxa are represented with distinct shapes and colors here that you can see. Potential interactions between species, for example, exchange of metabolites, predation, cross-feeding, etc., are indicated by lines connecting the taxa. These lines are drawn based on co-abundances of these species across a range of conditions or samples. Gray lines indicate positive co-abundances and may point to positive interactions, while yellow lines indicate negative co-abundances and putative negative interactions. By using this approach at a community level, taxa that are critical to the community can be identified. These may be species of high betweenness. Okay, so that is acting as a bridge point connecting two separate clusters of taxa, or high degree, having a large number of connections to other taxa. So in an ecosystem, these evolve mutual dependencies, symbiosis, and so on. So positive co-abundance interaction here between these, and then you have negative co-abundance interaction. So you have smaller number here, high betweenness species here, because it's sitting between high degree species here and this one here, and it's got high abundance in that sense. And you have a dead end kind of thing here. So these do not have high connectedness in that sense, high degree of connectivity, right? Whereas these are. So this is just a kind of a schematic, but you get the idea. These are kind of interesting, so I don't know how many I want to read, but let's just go ahead and look at a few more. So isolate engineering with fabricated ecosystem and community gene editing, high throughput genetics, and ecological containment. So rhizosphere and bulk soil. You want to have good bioengineering, but you don't want things to get out of control either. Okay, so I won't read all those details here. And this is now genome editing approaches for soil microbiome engineering. So you have site-specific recombinations. So that's a genetic, recombinations happen naturally during sexual reproduction, but this is slightly different. And you have homologous recombination, so that relates to chromosomes and how they are heterologous or homologous, so same gene. I won't go into the detail there. Conjugative transfer, so you have the CRISPR words being used here, and here is CRISPR Cas transposes, so obviously very technical things that I am not at all going to get into, but you know that genome editing approaches are now very common. They are just being considered here in the context of soil microbiome engineering. CRISPR is now very common. They are growing trees, for example, with edited genes that can become sustainable for paper production. The amount of paper we use, just even toilet paper, is so large that it's not sustainable to keep cutting real forests. But if you have genetically engineered trees that can grow again and again, and other ways of sustaining biomass or feedstock for paper, then you can really make this business sustainable, as opposed to trying to get people to switch from, let's say, toilet paper to a bidet where you use water instead of paper, right? So I will jump straight to the end. Remaining gaps and opportunities for soil microbiome engineering. Although this review has summarized the current state of the science, several hurdles remain before wide-scale implementation of soil microorganisms can be applied for tackling pressing issues in the face of climate change, an increasing global human population, and a decline in wildlife habitat and biodiversity. So motivation is not really needed as to why we need sustainability of soil microbiome, and sustainability of food production, food security, water quality and quantity, and so on. Soil microorganisms have the potential to alleviate issues associated with these challenges, but we are still largely working in context with several unknowns with respect to understanding the functional potential of soil microorganisms, how they work together as a community, and how they can best be collected and formulated to provide the desired beneficial outcome. Furthermore, with respect to working with genetically engineered strains, regulatory hurdles remain that limit the widespread application. Especially in a country like India, there's lots of debate about genetically modified organisms, cotton and brinjal, eggplant, and so on. That farmers want to use them to make money, but there are lots of concerns, especially from let's say activists who think all GMOs are bad and so on. The largely untapped potential of soil microorganisms presents a tremendous opportunity for mitigating many of the challenges facing the environment. These opportunities include using natural or engineered soil inoculants and consortia for remediation of polluted and otherwise degraded soils, maintaining and improving crop performance, and mitigating certain negative consequences of climate change. These are the opportunities. For example, soil microorganisms may help to reduce levels of greenhouse gases and sequester carbon in soils, which is very critical and has a virtuous cycle of increasing soil nutrients, soil moisture retention, drought resilience, and so on. Advances in genetic engineering of soil microorganisms and associated knowledge of biocontainment of released soil microorganisms can also be valuable for containment of unintended releases or cases of bioterrorism where intentional releases are made. Lastly, knowledge obtained on how best to manipulate soil microorganisms for desired functions may be used at a systems level by soil managers for specific crops by adding soil amendments that optimize the chances for natural soil microorganisms to grow and thrive. This last situation may turn out to be one of the most valuable for taking advantages of the inherent beneficial properties that are carried out by interacting members of the soil microbiome. Ultimately, the combination of several strategies is necessary to meet the challenges that we are facing with climate change and a growing human population, including genetic engineering of microorganisms and plants and use of best practices that promote ecosystem sustainability. Well, you can imagine someday we may have was that the last slide? Yeah. We may have, instead of throwing chemicals like fertilizers, we may just be throwing a can of microbes into the soil to make it all healthy. This happens, for example, to chew up oil spills. There are bacteria that can eat up the hydrocarbons. The oil is basically organic matter, so that kind of solution, biotechnology solution, may well come out to be sustainable, workable, and without negative impacts. So let me leave this here and we'll move to the next topic. I don't know what it is, but maybe I can guess. Let's see. It could be model hierarchies. A lot of people talk about model hierarchies for understanding phenomena like the monsoon. So let's come back and discuss that in the next podcast.